Well, I think we'll go ahead and get started. You're here for the crop seminar. You're right. So today we're going to have Uta McKelvy. She's a new doctor, just got her doctorate degree in pathology, and she had been here before at the GN. And I tried to convince her that extension would be a good place for her to be. So we created a little bit of room with some moving around of pathology people on the extension side. And so you can tell them what your appointment is, but she's with extension. Then Dyer has been here a couple times as well. He'll be speaking secondly. She's going to speak about foliar and root diseases. And then he's going to talk about seed treatments. And Alan has been here before, and he has a pathology appointment there in the research side at MSU. So as always, when they get offline, you're stuck with me. So ask questions of the experts while we have them. So go ahead, Uta, you want to go ahead and start?
Yeah, I can do that. Let's see.
Just one thing, one program note. If you're online watching, if you can make sure you're muted and turn your video off, that'll help our streaming. And talk with me afterwards in the chat box so that I know you're there and I can give you points for the pesticide program. Go ahead, Uta.
Alrighty, I hope everybody can hear me and see the screen. I assume since it worked before, it will work now. Yeah, so good afternoon, everybody. Thanks for the invitation, Marco. It's a pleasure to virtually join you in Malta. You know, I actually thought earlier today that the last time I got to leave Bozeman was February last year when I came to Malta for the last extension talk. And ever since I've been stuck on campus or at least in town. And so, well, I hope, you know, next year around this time we can meet in person again. But for now, we'll make do with what we have. And so I thought I'd talk today about a couple of common pathogens that cause root and crown rot diseases, preferably or mostly in wheat and barley. But I also want to talk about pathogens that we commonly observe on the foliage of those cereal grains, and then also on the head and the grain itself. I guess I should mention my title, since Marco already mentioned that I recently got my PhD, so I get to call myself now Dr. Uta McKelvy, and my appointment is officially Associate Extension Specialist, meaning that I'm largely filling in for Dr. Mary Burrows' extension responsibilities since she moved on to be the director of the Agriculture Experiment Stations. So with this coming season, if you have any questions or concerns regarding what might look like a plant disease in your field crops, feel free to reach out to me. That's why I included my contact information here. Or you can also reach me through the Scudder Diagnostic Lab, where I'll be working as diagnostician come the season. Okay, so I'm probably not telling you anything new if I'm revealing to you that there are plenty of diseases that can plague our crops, our cereal crops in this slide specifically. And those diseases can affect the root and crown, as well as the foliage, and then also the head and grain. And then there are also viral diseases that we consider systemic, so they infect the entire plant. And so, I mean, obviously these are things to worry about, but I don't want to worry you today that I'm going to like squeeze all those diseases I'm listing here in the next 50 minutes. So I'm going to focus on a couple of diseases that are very prevalent in Montana and are reoccurring every year. And you're going to learn which ones those are in the next couple of slides. So my objective for this talk today is to talk about these common fungal pathogens and refamiliarize you with the symptoms that they commonly cause on the crop, which might help you identify what issues you're observing in the field. And of course, I want to give you some pointers with respect to how to best manage those diseases. But what I also thought to do is I kind of want to point out some connections to you, for example, between pathogens that affect roots but can also reoccur as foliar diseases and affecting the grain. And I also want to give some food for thought as to what does a pathogen do once one crop is harvested and another one goes in the field? Where does the pathogen go? So we kind of start thinking about pathogen movement or persistence throughout several crop rotation sequences. And of course, I wanted to make sure that I can supply you with many helpful resources where you can find more information or just recall the information that you may have, missed today. So before we delve into the specifics of some of the diseases I selected for today, I thought it's a good idea to start with a disease 101, which is the disease triangle, which is shown here on the left. I hope you see it on the left. So the disease triangle tells us that in order for disease to occur, there has to be a pathogen present, and this pathogen has to meet a susceptible host. And then in addition to that, environmental conditions have to be favorable for this pathogen to infect the host and spread and for disease to occur. So if either of those three components is missing, you will not observe disease. And so this is important to keep in mind because understanding the factors that go into each corner of the disease triangle can be very helpful for us in finding out what disease we might be dealing with or how we can manage those diseases. And so when we were talking about pathogens, there's a whole host of pathogens that could possibly affect our crops. Namely, there are fungi, bacteria, nematodes, and viruses, to name the most important ones. But today I'm going to focus specifically on fungal pathogens of cereal crops because there's only 1 hour for me to talk. And I gotta have a reason to come back and talk some more. So we'll save the rest for later. All right, another important information for you. Fortunately, you're not alone in having to figure out what exact disease you're dealing with in your crop. Next to your highly competent extension agent who can help you with diagnosis, there is also a fabulous team of diagnosticians located at the Scudder Diagnostic Lab here in Bozeman, Montana. And we're here to analyze your samples. And in this specific situation, it's not only about plant diseases, it could also be insect pests that you might be observing, not just from your field, but also from your house or your garden. We're also looking at herbicide injury. We're diagnosing weeds or what might be weeds, etc., etc. And you can find all the information that you need on the website of the diagnostic lab, which I put here on the slide as well. Okay, so let's start with the diseases that I actually want to talk in more detail about today. So I thought I start, from the base of the plant and then work my way up. And it's also kind of a look at the progression of diseases throughout the season. And so I have two root rots, which we typically categorize as late season root rots that I want to highlight for you today. So the first candidate being what we call common root rot, which is caused by a fungus that is called Bipolaris sorokiniana. It also comes by a second name, which is called Gliobolus sativus. And so common root rot, as the name implies, is the most commonly observed root rot in Montana. The tricky thing with root rots is that by the time you observe symptoms in the above ground parts of the plant, the root rot has already well established in your crop. And so to find earlier season symptoms, you'll have to actually dig up your a couple of plants and look at the roots and crowns to determine if there is a root rot going on. So a telltale symptom for common root rot is when you dig up the base of your plant, you will observe a discoloration that is brown of the subcrown internode, which is the segment between the seed and the crown. And typically what we also observe with common root rot is these brown and shiny streaks at the base of the plant, which are shown here in this picture. And they rarely extend above the first node, so you will always observe them very close to the soil line. Generally, the root system is brown and discolored and somewhat reduced. Later in the season, you will notice this disease as heads that are maturing earlier than the rest of the field, and the grain may be shriveled or there may not be grain at all. Another widespread root rot or crown rot pathogen is Fusarium root, crown, and foot rot. And so this is actually a disease complex that is caused by several Fusarium species, the most prominent of which in Montana are Fusarium culmorum, Pseudograminearum, and Graminearum. And so what is typically associated with Fusarium root rot is you observe it more often in spring crops, so much more in spring wheat rather than in winter wheat. And this disease often becomes severe in years where we have fluctuating water conditions. So say a moist spring and then a very dry summer. And often this disease is also severe in fields with high nitrogen fertility or nitrogen content, excuse me. And so the reason for that being is that conditions like a lot of soil nitrogen and also soil moisture early in the season promotes ample vegetative growth of the crop. But then later in the season, typically around flowering or grain fill, we often have not as much precipitation, right? So the plants enter water stress, and those plants that have produced a lot of vegetative growth will enter water stress more sooner or more severely. And these are exactly the conditions where Fusarium root rot thrives and has a considerable impact on crop productivity. Some telltale symptoms of this disease are, as I mentioned before, these white heads. They're pretty typical for this Fusarium root rot in the field. And then when we're looking at the root system and the lower stem, typically the roots are brown and discolored, especially the subcrown internode, and rather poorly developed. And we will see this characteristic chocolate brown, dark brown discoloration of the lower stem, which extends up to the second node. And under very moist conditions, you even may observe some pinkish mycelial growth inside the stem or even outside the stem or around the crown tissue, which almost looks like cotton candy. And that is the pathogen growing out. Although these are different pathogens that cause these different diseases, generally factors that cause the disease are very similar. And so management strategies are also very similar for these two diseases. So namely, and I'm not going to talk in too much detail about this because this is what Ellen's talk later will be about, but seed treatments are very effective, suppressing an early infection of your crop and an early establishment of root and crown rot pathogens in your crop. Especially with common root rot, planting not too deep, so at about 1 inch deep in the soil, can be really effective. The idea here being that we're trying to reduce the length of the subcrown internode, which is the primary area of attack by this common root rot pathogen. And then, as I mentioned earlier, water stress situations are what promote root rots a lot. So anything that we can do to set the plant up for success and reduce their stress will also make the plant more resilient against root rot diseases. So this can be minimizing water stress, so meaning that we will plant adequate for what we anticipate precipitation and water levels to be in the year, and also fertilizing specifically with nitrogen adequate to what yield we're going for and how much precipitation is typically available in your location to support plant growth. And then, of course, choosing varieties that are adapted to grow in the Montana environment can be very effective, as well-adapted varieties are getting less easily stressed. And so which varieties those might be, and also other management recommendations, you can read up in this month guide, which I put up here in the top right corner about this month guide about small grain root and crown rot diseases, that talks about those diseases that I just highlighted and also gives you some management recommendations. And of course, one important management recommendation, which you will hear several times throughout today's talk, is crop rotation. So it's important to interrupt your crop sequence with some non-cereal hosts, which suppress or reduce the amount of pathogen present in the soil and thereby disease risk in following seasons. All right, let's talk about some foliar diseases. All of these foliar diseases, which is by no means an exhaustive list, have a couple of things in common. So for one, all of these foliar diseases that I'm talking about today, the pathogens are residue-borne, which means they're coming from the ground. They survive on infected stubble and plant material that remains in the field from the previous crop. And also all of these diseases perk up or pick up and show up in your field after when conditions are very moist, so when the canopy is very wet, which might be after several days of rainfall and warm temperatures, or if we have conditions where we have extended dew on the crop for several hours throughout the day. Okay, so two very commonly observed foliar diseases here in Montana are on the one hand tan spot, which is caused by this pathogen Pyrenophora triticirripentis, or someone else might pronounce it differently, so I prefer to just read these names to myself, but now I said it. Okay, and the other one is Septoria leaf spot caused by Septoria tritici or tritici, or it's also called Staganospora nodorum. And so these two diseases look pretty similar, and you have to kind of have a trained eye and look for specific things to be able to distinguish between the two. So tan spot, what is characteristic is this eye-shaped lesion on the leaves. And sometimes these eye-shaped lesions have like a brown pupil in the center. What is very typical for tan spot is this yellow halo around this tan colored lesion, which is a result of toxins that this pathogen produces. On the other hand, Septoria leaf spot is more brownish, I would say. And the lesions are more diffuse, and they can also coalesce into bigger patterns on your foliage. And both of these diseases, or both of these pathogens, if conditions are favorable, meaning a lot of moisture and nice warm temperatures, they can spread within the canopy onto higher and higher leaves, onto the flag leaf. And if conditions persist and are conducive, these pathogens can also cause what we call gloom blotch on the grain, which in the case of tan spot or the Pyrenophora pathogen looks like a pinkish discoloration of your grain, which we call red smudge. And in the case of Septoria, this is we see more like a yellowish brownish discoloration, and then these characteristic brown pycnidia on the grain, which is called gloom blotch. There are also a couple of foliar diseases, obviously, on barley. The same general descriptions for these pathogens when it comes to conditions that favor disease development apply here too. Especially, we observe these diseases in systems where we continuously crop barley, so there's little diversity in the rotation. And obviously, systems where till is minimal promotes the disease as well. And then of course, if we have malt barley, for example, that is irrigated, this irrigation again increases moisture and promotes these foliar diseases. There is on the one hand net blotch, which comes in two forms. The net form, this is like a serious tongue twister for me. So I find that a very good exercise. So we have the net form net blotch, and we have the spot forms net blotch. I'll say that really fast for practice, which are caused by two different special forms of Pyrenophora teres. And then there's also the spot blotch on barley, which is caused by a pathogen which may sound a little bit familiar to you now, Bipolaris soronciniana, which is the pathogen that I earlier mentioned can cause common root rot in wheat and barley, etc. So this pathogen can also reoccur, causing these spot blotch diseases on your foliage. And there is a third disease that is commonly observed, which we call scald, which is very characteristic in its appearance of these 10 almost see-through like lesions that are surrounded by a dark brown margin. And then, as I mentioned before, all of these foliar pathogens can cause gloom blotch on your grain if conditions are favorable. So while for us diagnosticians specifically, differentiation between these different pathogens is important, it may in the larger scale be not as important for you, since management of all of these foliar diseases is largely the same. However, when we talk about specific fungicides, etc., knowing which pathogen is specifically dominant in your area might be helpful in choosing the most appropriate fungicide, which I'll talk about in just a little bit. But so generally, management of these foliar diseases, here again, I have to mention crop rotation. So the more diverse our rotation sequence is, the more time we give, or the more we reduce the pathogen load in the residue, and the more we reduce the disease risk for foliar diseases. And then there are several varieties available that have varying, resistance traits for one or two of these foliar pathogens. So this is where knowing which pathogen is dominant in your area might be really helpful in selecting a good variety as well. And then any management practice that reduces humidity in the canopy, such as reducing irrigation schedules so that irrigation occurs less frequent, or decreasing seeding density so that the canopy is less dense. These can be practices that kind of manipulate the environment in the crop in a way that is less conducive for the disease to develop. And then if it's possible, light tillage that buries infested residue helps break down this residue, and also prevents these pathogens that survive on residue between crops to not produce these spores, which are then used for infections. So tillage can be really effective. And of course, if disease risk is high and this foliar disease really threatens your flag leaf and your grain yield, then fungicide use of a fungicide might be helpful. And so the next following slides, I want to talk a little bit about some considerations and some information that we found that might be helpful for you to decide if and when a fungicide use might be warranted. So there was a study conducted in North Dakota and Montana in grower fields and on some of the research centers in the state. The study was conducted over several years, and a total of, I believe, something near 150 fields of hard red wheat, both spring and winter wheat, were observed. And so those fields were generally all categorized as low risk for foliar diseases. And so fungicide, different kinds of fungicides were applied either at Feeks 2 to 3, so at the tillering stage, or later at Feeks 9, which is at the flag leaf stage. And what the researchers did was they were looking at whether the fungicide application actually resulted in a yield benefit. And they did some estimations under what conditions or at what yield gain a fungicide application is actually economically feasible. And so this slide here that I'm showing here is a result of the study. So what you're seeing here is these bars here represent the 32 different trials of 13 spring wheat and 19 winter wheat fields where fungicide was applied at tillering stage. And so on the left axis, it is indicated the yield response, meaning the difference in yield between those fields that were treated with fungicide compared to those fields that were not treated with fungicide. So essentially the yield gain from fungicide treatment for each of those fields. And on the left hand side, or actually this is right, sorry, I'm really challenged by directions. The right hand side, we see the net revenue. And so that was calculated based on an average wheat price of 22 cents per kilogram of wheat. And so the differences here that are shown, which are the cross and the diamond and the triangle, are the lowest, average, and highest fungicide application costs. So to kind of get a range with if fungicide application costs are low, how what is the net return for this and this yield gain from the fungicide application. And so to summarize this graph, essentially 72% of these trials, so about 2/3 of these trials had a positive yield response. So there was a yield gain as a result of fungicide being applied at tillering. However, only 50% of these trials had a positive net return at a midpoint, at an investment at midpoint value, meaning the average or the medium fungicide application costs. So meaning that not every trial where we have a yield gain actually represents a net gain where you're coming out with a plus at the end of the day. And so based on these data from the tillering fungicide application study, the break-even yield response would be 70.6 kilograms per hectare. So you have to have a yield gain of 71 kilograms per hectare or more to at least economically justify your fungicide application or come out with a net gain. Or alternatively, based on these observations, you have to have a wheat price of at least 26 cents per kilogram to justify a fungicide application, assuming these medium application costs of $15.32 per acre. Okay, so this graph is set up in a very similar way, only now we are looking at the results from fungicide applications at flex stage, so at FEEX 9. And so a total of 41 trials was looked at, 17 spring wheat, 24 winter wheat. You will notice here that the application costs for fungicides are a little bit higher than in the previous slide. And so the calculations for net gain, etc., are based on these costs indicated here. So under this scenario, 68% of the trials actually showed a positive yield response, but also only 32, so only about a third of those trials had a positive net return on investment. So although the amount of trials where we actually had a yield gain as a result of fungicide application was similar to the application at tillering, the risk of making a fungicide application that does not result in an economic benefit is actually lower for these applications at FEEX 9 or at flex stage compared to tillering. And so based on these observations here, you would have to observe a break-even yield response of about 150 kilograms per hectare to justify economically your fungicide application, or alternative, a wheat price of 32 cents per kilograms. Now, I guess one thing to discuss here a little bit would be why is the net return for the flag leaf fungicide application lower across, or why do we need to observe an even higher yield gain to justify that? And I think the situation here is that for one, when we apply fungicides at flag leaf stage, then the application costs are higher. And the other thing is that, as I mentioned, all of these trials were conducted in low disease environments. So, while we might observe foliar pathogens on, the lower canopy leaves, rarely are conditions conducive for these foliar diseases to actually move onto our flag leaf, which is the most important one for yield and economic gain. And so I think that an application that is more cost intensive at flagleaf, where disease is even less prevalent that maybe at tillering, I think results in just a lower risk or a lower likelihood of an economic gain. So lots of talking. Here's the take-home message from this whole situation. In the Montana environment, rarely do fungicide applications in low-risk environments provide an economic benefit. And so for you to make the decision, is this fungicide application worth my time, worth my money more than my time, or not, consider some additional factors that influence disease risk. So this goes back to the disease triangle and also to those things that I mentioned when it comes to management of the disease. So if you see early symptoms of foliar disease and you're wondering, is this going to be a problem at flag and will I need to prepare to spray? Consider what variety have you planted and do you know if it is resistant or at least tolerant to the specific foliar disease or pathogen you're dealing with? What are the environmental conditions? Have they been conducive lately to promote disease? So has it rained a lot? Have I seen a lot of dew on my foliage that would promote the pathogen spores to germinate and infect? And how early in the season do you detect this disease and how fast does it spread? So these are all things that can help you in this decision process. And another thing that I want to point out here to you is this fungicide decision tool that has been developed as a result of the study that I just introduced. And so I'm just showing a little snapshot of what it looks like. You can find this tool if you're following this link, or it's also referenced on the website of the Southern Agriculture Research Center in Huntley. And so I've just put in some, parameters that seemed more or less reasonable to me. What is interesting about this tool is that you can also indicate the cost of, applying your fungicide by drive down, which you can kind of adjust considering whether you're applying an herbicide parallel to your fungicide, etc. But so for a fungicide application cost of $15 per acre and with a yield gain of 5 bushels per acre resulting from this fungicide application and drive down effect of 2.2 bushels per acre, you'd have to have a wheat price of more than $8 per acre to justify your fungicide application economically. And I checked this morning what the wheat prices are. As of yesterday, wheat sells for $6.7 per bushel. So under this scenario, fungicide application wouldn't be warranted. And so this is just one example of how you can use this tool to make decisions for you as we're headed into the season and foliar diseases might show up in your foliage. You can also find more information here in the month guide on fungicide use, which kind of discusses the similar factors and some more that could help you make these decisions on fungicide application. And since I've talked a whole lot about fungicides, you're probably wondering, well, what fungicides can I apply? And I will admit to you that I'm too new to this business to be able to memorize those active ingredients and those product names. So whenever I'm getting a question like that, my first move is to go to the extension plant pathology website where a bunch of resources are referenced, one of which being the table of fungicide recommended for use to control foliar diseases of small grains and also head diseases. And so this is just a copy of what you would find if you followed this link. So I encourage you to check that out for more information on which products might be suitable for you. Alrighty, I was like, what, did we talk about this already? No, we did not. I had a thought here. So I want to switch gears and I want to use these last remaining minutes to talk about Fusarium head blight, also known as scab, which is can be a challenging disease issue affecting the heads and grain of our cereal crops. And so there's a whole host of pathogens that can cause this disease. And the reason why these root rot pictures show up here again is that some of those pathogens that we know to cause Fusarium root and crown rot are also pathogens that cause head blight. So Fusarium graminearum, Pseudograminearum, and Culmorum are pathogens pathogens that cause head blight. And then there is a fourth one, which is Fusarium avinaceum, which is also a prevalent pathogen that causes this head blight. In fact, a survey looking at which Fusarium species are causing Fusarium head blight in Montana, it looked at extracted 35 different isolates that were then later identified, and the location for this was in northeast Montana. And so what this study showed, or this survey showed, is that actually in Montana, the most dominant Fusarium species that causes Fusarium head blight is Fusarium avinaceum, and not Graminearum, which was previously assumed and described. So I'm going to leave that hanging here a little bit, and I'm going to come back to that later, and we'll recall this information for you. But before we dive into the specifics or some more complex aspects of Fusarium head blight, I thought I, share some pictures with you of what that actually looks like. So Fusarium head blight is a concern because obviously it reduces yields, and depending on the conditions and how much of the field can be affected, up to 74% yield losses have been described in the literature as a result of Fusarium head blight. It damages the crop in several ways. So for one, it can cause sterility of infected spikelets, which will then not produce any grain, or the kernel size can be reduced. And then these kernels are typically shriveled and discolored, which you can see here on this right-hand picture. We call these kernels tombstones, or as I will refer to them later, FDK for Fusarium diseased kernels. So they're smaller and kind of paler in size and just kind of sad looking, and obviously the test weight of those is also much reduced. Some other, another important thing about Fusarium head blight is that it not only reduces kernel size and kernel quality, but this pathogen can also produce several toxins in the grain, which are not good for humans, but also not for animals. So the most prominent of those toxins is called deoxynivalenol, or in short, we call it DON. And so there are actually thresholds for this toxin of 1 parts per million for grain that is dedicated to human food processing, and a slightly higher threshold of 2 parts per million for grain that is used for feed. In malt barley, there's actually a 0 tolerance for this toxin, as it is really interfering with beer production, which, we don't want to mess with that. So this is the impact of this disease in a larger scale, but what does it actually look like in your field? So what you might observe are heads that are appearing bleached, and maybe contrary to those bleached heads that are associated with the Fusarium root and crown rots, these heads bleached as a result of head scab are typically just sections of the head. So certain segments or partial bleaching is what we observe. And often we can see a discoloration at the base of the head, which you can see here in this picture, how it's like darker brown just below the head compared to this other matured wheat head. And under conditions of a lot of moisture around grain fill and grain maturing, we can actually observe the pathogen growing out of the infected sections of the head, which will then, have a pinkish to orange color or mold on the grain. Right. Okay. So what is the disease cycle? And this is really important to understand how we can manage this disease. So the one thing to know is that Fusarium head blight pathogen is introduced into the field through infected seed. So again, seed treatment and good quality seed is a very important factor to keep in mind for avoiding or suppressing this disease. But the pathogen can also enter the field through wind-borne spores, so spores that are blown in with the wind. However, in many fields, this pathogen is already present, and it can easily survive on residue that has been infested before. And so every, crop rotations that are have frequently cereals in it, so wheat, barley, durum, but also corn, really increase the presence of this pathogen, or can increase the presence of this pathogen in the field. And so this pathogen readily spreads by rain or wind. So from the residue on the ground onto the foliage and later onto the head. And so environmental conditions that are moist, so a lot of rainfall and dew, but also irrigation, if that is something you do, can really promote spore production and infection. So it's very similar in that sense to the foliar diseases. However, Fusarium head blight affects the heads and the grain of your crop. And so your crop is most susceptible for infection with the scab pathogen during anthesis. So right before, during, and after anthesis. And so if we have pathogen present in the field, and then during and right after anthesis, we have a lot of moisture, for example, rain, we run a risk of Fusarium head blight occurring later in the year. The tricky thing with this disease is that we will only observe the symptoms at soft to hard dose stage, so weeks after the infection actually occurred. So that makes it really tricky to manage this disease because it requires a lot of careful observation and anticipation. So I'm putting here this this graph of corn acres in Montana over the past 20 years. Because corn is often referenced as a potent source that promotes Fusarium, the Fusarium head blight pathogen. So because it is it's a good host for this pathogen. And so if we have a rotation sequence where we have corn that is then followed by wheat or barley, that significantly increases the risk of Fusarium head blight. And so Fusarium head blight has been more frequently observed in Montana over the past 20 years, and there is a likely association between the increase in corn acres as well. However, I will say last year we did not receive a lot of Fusarium head blight samples in the diagnostic lab, so I guess last year was not that bad of a year overall. There are similar management strategies to previous diseases, but then there are also a couple of management strategies that are a little bit different. So crop rotation, as I mentioned before, everything that is a non-host crop to these Fusarium species or the the head blight pathogens can reduce the risk or the pressure of this disease in your field. And then choosing varieties that are tolerant to Fusarium head blight can really be helpful. To prevent this disease from happening or have a big impact on your crop. Tillage practices that bury the residue and help break it down can be very effective. And then one management recommendation kind of goes along with how this infection occurs. So I point out here uniformity of heading, which you could achieve by increasing your seeding rate so that you have more primary tillers and less secondary tillers. And so then those primary tillers all mature or, head and flower and mature at a rather similar time or more synchronized. And so the thought here being that should it turn out to be a year where Fusarium head blight is likely because of moisture conditions around flowering, the difficulty if you do not have uniform heading will be when do you apply your fungicide, because there is admittedly a very narrow window for this fungicide application. And so having a crop that develops at a rather synchronous pace will, just achieve a better or will make your fungicide application more effective, or in other words, means that you will not have to apply fungicide twice, but just once. If you're doing irrigated production with barley, for example, you want to consider suspending irrigation during the time of flowering until early grain fill to really reduce that risk. And as I mentioned before, should it be a year where risk is high, consider using fungicides, and I will talk a little bit about which fungicides and when are most suitable. Again, here a quick reference to our Fusarium head blight Mont guide, which details a lot of the information I'm talking about today, black on white for you to read up again at home. One thing that I wanted to point out, sorry, often it is also recommended to kind of increase the quality of your grain and reduce the number of infected kernels in your grain lot, is to increase your airflow of your combine during harvest to kind of separate these lighter fusarium diseased kernels from the rest of your grain. And so thereby cleaning your seed and just achieving a higher quality in your seed lot. And so while this management strategy makes sense for your immediate, economic return, Frankie Crutcher, the plant pathologist at the Agriculture Research Center in Sydney, Montana, asked herself this question, well, what happens with this Fusarium avineasium pathogen on these diseased kernels that are blown out and left in the field? And so the reason why she asked this question is because, for one, she found that Fusarium avineasium is this very widespread and common pathogen that causes Fusarium head blight. At the same time, we also know that Fusarium avineasium is one of the most prevalent pathogens that causes root rot of peas. And so if you have a crop rotation where your wheat is maybe, or your wheat or your barley is followed by peas, does the separating the Fusarium diseased kernel from your grain harvest remaining in the field, does that increase the risk of Fusarium root rot in your subsequent pea crop? And so this is exactly the study that she conducted. And some of those results I want to share with you because I think it's very interesting and it's worth considering for the future. So just to kind of give you a picture of what I'm talking about when I'm talking about Fusarium root rot in peas. So it's a big problem in pea production, obviously, and it causes plants to grow poorly and produce poor pods and seed. And so the cause for this is a decay of the root system and a discoloration, which you would observe if you were to dig up these pea plants and look at the system. So the question she asked herself is, if there is Fusarium avineasium in the grain, and then the Fusarium damaged kernels will be blown out by the combine, will this increase the risk of root rot in subsequent pea crops in years where we have high Fusarium head blight incidence? And so what she did is she conducted a field study over 2 years at 2 different locations in Montana, namely Sydney and Huntley, where she spread artificially Fusarium diseased kernels into fields in the fall at different concentrations, and then she planted those fields with a yellow pea variety AAC Carver, and she looked at Fusarium root rot in peas after 3 to 4 weeks after emergence of those peas. And so she estimated different concentrations of Fusarium diseased kernels based on what the Canadian Grains Commission estimates how much Fusarium diseased kernels occur in a grain lot of durum wheat and spring wheat. And so she did like some math about what was the harvest this year, how much grain would that be that is diseased, and she came out with this range. And so as I mentioned, she did a root rot severity assessment on these pea roots, and after 3 to 4 weeks after emergence. And so this is the disease severity scale that she used, just so you kind of know where this is coming from. It's essentially a scale that documents progressively more severe symptoms of Fusarium root rot disease. And so what she found in her study is that not across all locations and all years, but in 3 of the 4 location years that she did the study, she did find that the more Fusarium diseased kernels are left in the field, the higher the incidence of Fusarium root rot on peas in the subsequent pea crop. And not only was Fusarium root rot detected more frequently, which is what incidence describes, but also in 2 of those years, there was a relationship between the amount of Fusarium diseased kernels spread in the field and the severity of root rot disease that was observed in a subsequent pea crop. And so this is a very, very short, summary of a very extensive study that she conducted. But what are some conclusions from the study that I want you to kind of keep in mind and think about a little bit? So in the greenhouse, she could show that Fusarium erinaceum isolates that she obtained from Fusarium head blight fields were actually capable of causing severe disease in pea plants. And she did observe that increasing the inoculum of, so the amount of Fusarium disease kernels in the field increased severity and incidence of field pea root rot in two locations, it's in 2 location years in the study. But she did not observe, which I didn't show here, a yield or an effect on yield. And this might also be because, she observed these root rot severity at an early stage in plant development and then didn't follow up what that might look like at later points of the plant development. And so although there is no effect on yield, or we don't know how this pea root rot develops through later in the season, what needs to be considered is that the more often peas are planted in the field, the more Fusarium pathogens we are accumulating in that field that can cause progressively more root rot and so it's worth considering how much we are contributing to the pool of pathogens that cause pea or other pulse crop root rots by this practice of spreading these diseased kernels into your field. So if there is one take-home message for you to carry from this talk today is just kind of be mindful where you clean your seed, specifically for Fusarium head blight, or how you're disposing of those kernels and which crops are following that field and what impact it might have on those. Because, these studies that look at disease development over several years and sequences of crops are rare because they take a lot of time and it's hard to follow through. So we may not know as much about it, but I think this study is a good indicator that it is worth thinking about it. I'm going to wrap this up here really quick because as usual, I'm talking too much, but I guess that's an extension quality. So I carry it with pride. So I mentioned fungicide applications. They can be effective for Fusarium head blight management. But, I also mentioned it's not that easy to decide when to apply or anticipate if it's necessary to apply and when to apply. And specifically with Fusarium head blight, it's all about timing. For wheat and durum, early flowering is typically the time where you want to apply fungicide if disease risk is high. In contrast, in barley, you will have to apply this a little bit earlier at early full head emergence. And here are some pictures to indicate what your crop looks like at the ideal time for application of fungicides. And again, this is a reason why you want to aim for a crop that develops very homogeneously so that it will be easier for you to determine when is the right time for this fungicide application. And then another thing that is really important is to achieve a good coverage of all your spikes. So it's not, you don't want to just hit your canopy like flat from above. You want to have your spray booms at an angle to make sure that you get both sides of your head sprayed and covered with fungicide, because good coverage is key. And so I included some information here. It's just probably too much for you to read right away, but I do admit that I'm not very techy when it comes to spray booms and nozzles and stuff. So honestly, this information here is not telling me a whole lot. I hope it spark some thoughts in you, and so I'm gonna kind of skip over that. I will tell you it is included in the month guide on Fusarium head blight from Montana State University. A lot of research has been done on which fungicide products are actually effective in suppressing Fusarium head blight. And so one thing to point out here is that strobilurine fungicides, or those QOI fungicides, are generally not recommended to use for Fusarium head blight control, as they have been shown to increase the concentration of this toxin in the grain after application. DMI fungicides or DMI fungicides are the most widely used fungicides to control Fusarium head blight, and they are good at suppressing Fusarium head blight, but different active ingredients are, differently effective, which is shown here in this table. So generally, Prosaro, which is a combination of these two active ingredients, achieves a good control, and so does Caramba and Proline, whereas Folicur and Tilt, which are active ingredients, tebuconazole and propiconazole, are generally not having or achieving a very good suppression. So if you had to choose a fungicide, I would recommend these upper three. And again, those fungicides suitable for control of head scab are also listed in this fungicide reference table that I mentioned earlier, which you can find under this PlantPath MSU extension link. Okay, my last point, and then I'm done for sure, goes out to integrated pest management. And that is that, combining more than one management strategy of those that I listed for Fusarium head blight has been shown scientifically to increase control of Fusarium head blight. So there was a study done by Marsha McMullen in North Dakota who showed that applying crop rotation and choosing a tolerant cultivar reduced the risk or the severity of Fusarium head blight and winterweed by 80%. And applying a fungicide in addition to that reduced the severity by 95%, which is pretty significant. So it's all about, choosing those many little hammers to plug or to chisel on a problem. Is that the right word? Probably not. Rather than, relying on just that one management strategy and hoping that that will do the trick. And with that, I'm gonna mute my microphone here and let someone else talk, or just listen to your questions that you may have. And I'm gonna leave you with these resources where you can find more information on what I've talked about today. And that's that, and I thank you for your attention. Somebody's still awake? I'm not seeing anybody here. Alan is still here, that's good. Somebody listened. Awesome. Well, hey guys, now I see you. I just saw heads before. Are there any questions?
Have there been any studies just with showing the idea of just using grazing methods when you indicated the reduction?
Are you talking about Fusarium head blight right now, or what are we talking about?
Right, the idea of reducing the head blight by using grazing methods, fall grazing.
Well, so I mean, the one thing is with grazing is to consider that these mycotoxins that Fusarium head blight produces are of concern for animals. So I guess that's something to consider about. And then, the other thing with grazing is that it doesn't really reduce residue that remains in the field. So I think, actually, I don't know if studies have been done, honestly, but I'm not sure how effective that can be. Maybe Ellen has some thoughts on that since no, you don't. Okay.
No, that's a great question. I don't know of any study done on the grazing, and I have had growers ask that before. Just don't know. I think, people have asked also about silage in corn, doing silage versus grain corn. And I just don't think there's any good information on the impacts of those practices.
Yeah, no, studies are always, a little harder, or a lot of them are always a little harder to conduct in Montana. From personal experience, when I really focus my attention on a disease, it tends to disappear. So maybe it's time to really work on Fusarium head blight to do you all a favor. I don't know. I had the luck with my dissertation thesis where I was working on wheat streak mosaic virus, which then just vanished from the landscape for the past couple of years. So. But so it takes a lot of time, and you have to have a lucky streak to hit some years where you have good disease to be able to kind of see what your treatments, how effective they can be. And so that's often challenging when it comes to, taking studies into the field and gathering data that are important for you growers.
Questions for Uta? So I gave you a little evaluation tool. She's trying to improve on her skills. One thing I'll say, it wasn't long ago where Uta only understood German. So she's come a great way and has a little bit of humor, which we all need in her presentation, as well as good information. So thank you very much. Appreciate it. Good job.
You're so welcome. And if there are questions occurring after today, when you had some time to digest all of this information, shoot me an email, give me a call. I'm here to help and I'm excited to do so. Okay, with that, I'll mute myself and stop sharing.
I don't know if I can move the sharers. That's not me. Okay, Alan, you want to go ahead and, well, do you guys want to take a second?
You want to take a break?
Let's take a 3-minute break.
Okay.
Dr. Dyer.
Are we ready?
Yeah, so as I said, Dr. Dyer has an appointment as a pathologist. You can tell a little bit more. He does some teaching and some research and even some field trials. We tried to help him out with some of those here about a year ago. And he's been up here a couple times, and just really appreciate him being interested in helping growers and not just the textbook side of the pathology question. So he's going to talk about seed treatments. So thank you very much, Dr. Dyer.
I don't know who you're talking about when you say doctor. You can just call me Alan. You know, I really appreciate being asked up again. You know, it's always I'm a little rough around the edges socially. And so, you know, when I give a talk and I never get asked back, I wonder what I said. But anyways, you guys have been pretty tolerant of me. Anyways, what I want to talk to you guys today about is fitting seed treatments in your production system. And so let's just get this thing working here. What I will focus on, the goals today is maximizing your seed treatment, particularly talking about disease targets that are good ones for different systems, practices that will enhance seed treatment efficacy and application processes. I also then want to talk a little bit about the benefits of seed treatments, and then you guys have an opportunity to run for the hills. I am going to put an ask out about a grower participation research project, kind of filling a need in terms of information on seed treatments. So let's talk about maximizing your seed treatment, particularly targeting. This is not a comprehensive presentation. There is a small grain seed treatment guide out there that provides more comprehensive information about particular products. But what we're going to do today is we're going to target our conversation on particular issues. The primary targets of seed treatments are the smuts and bunts, seed and seedling diseases, and the root and crown rots that Uta mentioned earlier. The first thing that I want to talk about is the smuts and bunts, and these diseases cause problems with yield as well as issues with taste and making malt. The two most common smuts and bunts affecting wheat in Montana are dwarf bunt and loose smut of barley. Those are the ones we get 2 or 3 reports of uncontrolled dwarf bunt and loose smut of barley, and those cause considerable cost for those growers that have it. Sometimes people will also get common bunt. Dwarf bunt and common bunt are also referred to as stinking smuts, because if you have them in your field, they will impart a rotten fish smell to your grain. So that's not so good when you're going to the elevator, and you can smell it quite a ways away. I have a small 4 by 10 foot plot a couple hundred yards away, and we're harvesting it. I can smell it from across the section. So it's pretty potent stuff. Dwarf bunt is exclusively a winter wheat disease problem. It's requires 90 days of snow cover for infection of the plants. Infected plants will have flared-ons. This is the same as with common bunt. And then you have the grain replaced by what are called bunt balls, which are just masses of these stinking smelling spores. The issue with dwarf bunt is that not just the fact that it reduces yield and produces these stinking spores, but also this is a phytosanitary issue restricting trade, particularly for seed, but also for grain shipments to other countries. So we do probably 30 to 40 phytosanitary certificates where we inspect grain going out for international shipments for this. There is some moderate resistance in Yellowstone and Flathead to this disease, such that if the disease pressure is not too high, you're going to be okay in the domestic markets. If disease is high, though, those things will fail. A variety called Clearstone is supposedly resistant, and I haven't tested it, but that is line developed by Phil Bruckner as well. Phil's done a great job with those. What really is the best practice for controlling dwarf bunt is application of a seed treatment called difenconazole, or used to be called Dividend. Highest rates of difenconazole virtually eliminates this disease from fields. There's no risk if you're applying high rates of this, of you having a problem with dwarf bunt. One issue with difenconazole is that some cultivars are affected by that high rate of difenconazole, and there may be a delay in emergence with it, such that full emergence is delayed by 1.5 to 2 days. And so, you know, if you have a half your field, you use something that didn't have difenconazole and then you had difenconazole, you'd be like, what the heck? But we haven't found that little bit of a delay has any impact on yields. But it does certainly make you a little concerned. The other most common smut and bunt is loose smut, barley caused by Usta nuda. This is a seed infecting smut. If you have a problem with this in your field, what you're going to notice is these naked rachi. So you'll have the florets are completely replaced by spores, and those spores will be gone by time the harvest comes. So they're not going to affect, those spores really won't be in your harvest. But the problem is those spores have already gone over to the neighboring uninfected plants and they infect the seed. So this, we have 2 grains, kernels of barley here in the bottom. The one on the left, both are stained for mycelium, and the one on the left is uninfected and the one on the right is infected. And you can imagine that grain on the right is not going to make good malt, and it also is not so palatable for cattle and things like that. So it's something that you have to watch out for. People often like to keep the same seed treatment for their farm, right? So you use the same thing across everything. Well, if you got winter wheat and you're applying difenconazole and you think it's going to control this disease, it doesn't. Difenconazole will work on loose smut of wheat, but it doesn't work on loose smut of barley. It's ineffective. There are many seed treatments out there that will provide good protection, but not perfect protection for this disease. The most important part of protection is clean seed, because that's where the pathogen is coming from. Any questions? Guys doing good? Okay, I want to talk about seed and seedling diseases. Oops. The seed and seedling diseases, we have one seed disease, dry seed decay. This is also a problem in your bins. This is primarily associated with moisture issues, and we'll talk about that in a minute. And then we'll talk about the seedling pathogens, the Pythium, the Rhizoctonia, Fusarium, and Cucullabis. Dry seed decay is something you'll see in winter wheat. In the field, we'll see with winter wheat, and it's associated with production of mycelium on the kernels. It will have a greenish appearance, or if you're checking your field and you have very poor stand and you go out there and you pull up these kernels and they're just, the soil just sticks to them. Well, they're sticking to all that mycelium from the fungus. This is for in the field conditions. Dry seed decay is primarily an issue for winter crops associated with planting into a dry seed bed where the soil moisture is 13% or lower. And what happens is that seed becomes moist enough for the fungus to grow and attack the seed, but it's not moist enough for the seedling to protect itself. So it's not the seed itself is not biologically active. And so the fungus is an opportunist and comes in and takes things out. And penicillin and aspergilla are everywhere. And so it's something that we have to watch out for winter wheat. I don't know how many, I don't think too many of you in Phillips County are with winter wheat. What people sometimes do is because they want to plant, you know, you lose your patience and you want to plant early because your yield potential goes down as time goes on. And so people will try to plant to moisture, and that's fine But if you're going below 2 inches, you're going to have some problems, and I'll talk about that in a little bit. And the other thing some people do that I don't recommend is they will produce a deep furrow that's about 4 inches deep to get down to that moisture so that they can do a planting in late September. And both of those practices are fraught with other problems, and I'll talk about those later. What I recommend, generally speaking, is to wait for moisture, and that's a hard thing to do. If you're kind of losing your patient, there are seed treatments that will provide 2 weeks of control. Almost all the seed treatments that are registered for dry seed decay will provide about 2 weeks of good control. So you have a 2-week window there. You plant, and hopefully that moisture will come in. I will say this product called a Mausolil, it's off patent, is actually the best product for dry seed decay. And it probably will go off registration in the next few years. But if you're a winter wheat producer and sometimes you lose patients, which I do, I did with my 4-year-old this morning a little bit, and Mausolil is a good product to consider. Okay, so any questions about dry seed decay? Okay. Seedling pathogens come into I'm putting them into 2 categories. We have the Pythium and Rhizoctonias, and we have the Fusarium and the Cochleobolus seed pathogens. And the Pythium and Rhizoctonias, I'm putting separate because these pathogens are able to attack one seedling, and then they use that resource from that one seedling to go over and attack another seedling. And that means that they have this ability to have plant-to-plant spread. Fusarium and Cochleobolus don't. What that means for us is the disease caused by Pythium and Rhizoctonia tends to be aggregated. And that means that you tend to have no neighbor to take advantage of you being missing, right? So if you have diffuse disease and you lose a plant, the neighbor takes advantage of the nitrogen and phosphorus and moisture that you didn't use and produces even more grain. So you don't have as big a yield effect in that situation. But when you have a disease like Rhizoctonia or Pythium, this aggregation of disease means that the yield effect per loss and stand is higher. And so these are diseases that I want to particularly take control of. So I'm popular today. The other thing about this is using. Hello, Mike. I'm in a meeting right now. Oh, okay. Sorry. The other thing about this is that if you increase seeding densities for Fusarium and Cochleobolus to take advantage of the fact that you're not getting the stand you want, you can just increase the seeding rate, right? Because you're not going to get plant-to-plant spread. But if you increase your seeding rates to get that stand you weren't getting because of Pythium or Rhizoctonia, you're actually going to increase the problem you have. So it's pretty critical that these diseases we try to control. Hope that makes sense. Where do we see these 2 diseases? Well, Pythium seedling blight is most commonly associated with cold, water-soaked fields. Sometimes, particularly Pythium phanodermatum, it could also be in high warm conditions, but all the cases, it's water-soaked fields is when Pythium attacks. It's called a water mold for good reason. Sometimes it will not cause loss of stand. It will cause the plants to have an early season yellowing that the plants can grow out of. So you often have some agronomists say that yellowing is due to nitrogen deficiency. And in a way, it probably is, but I think it's probably Pythium. The Pythium is killing off a bunch of the roots, and so the plant is not able to acquire that nitrogen as well. And so, but usually they grow out of that. This disease, we see the best responses from seed treatments for controlling this disease for winter wheat, durum wheat, barley, and irrigated production. Spring wheat most times tends to escape this disease. Practices that people use for controlling Pythium include, not you guys, not for wheat, but for sugar beets, they'll use raised beds or rows. And for you guys, something to watch out for: phosphorus nutrition for all these seedling diseases. Make certain you have good phosphorus, not over phosphorus nutrition, but good phosphorus nutrition is really good for managing seedling diseases. Seed treatments that are effective for Pythium are the mefenoxam and metalaxyl products. These products have been there for 30 years. And so some of the cases, particularly if you're in irrigated production and you've been using this for a long time, you may have built up populations that are resistant to these products. There is a new product on the market called ethaboxam, and this is an alternate product, but it's not a panacea, and I want to tell you why. When metalaxyl or mefenoxam were first put on the market 30 years ago, those products were universally effective against the Pythium species. Ethaboxam, in contrast, this here is a diagram identifying particular species that are resistant to ethaboxam, and here is right here in the middle. All these guys that are in orange are resistant, and right here in the middle we have the Pythium F. anadermatum. And so I've done trials with this product, and I use Pythium ultimum, which is susceptible to it, and 3 years out of 4 it works great. But that 1 year I have one of these alternate species and I don't get any benefit from it. So it's something that's, if you've been using metalaxyl a long time and you're in a high disease situation, it might be worth checking out, but it's not a panacea. Rhizoctonia bare patch is the other big monster on the midway in terms of seedling diseases. And this pathogen, one of the classic symptoms for it is that it causes this spear tipping of the main roots going to the crown of the plant. It prefers warm temperatures, soil temperatures of 70 degrees Fahrenheit, and wet conditions, causes spear tipping, and it spreads from, it's a monster in terms of ability to spread from plant to plant. Where we see it most severely is when people plant soon after they apply a glyphosate treatment. And I'll just show you a picture of that in a minute here. There's numerous seed treatments that provide pretty good control for Rhizoctonia. Unfortunately, there's no studies having been done on whether these seed treatments will prevent the damage caused from planting relatively close to glyphosate treatments. This is a study done by Dick Smiley, and what he found is, and this is not something I can ever recommend to you guys, but that the waiting from the time of applying glyphosate to control a heavy volunteer load to planting your seed does provide a yield benefit for barley production, such that we're getting approximately 700 kilograms per hectare increase in yield by waiting 3 weeks relative to planting immediately after glyphosate treatment. This here is, I just put this up here just to, I don't know if the entomologist would be here today, but this is in the Moxon field station. This is entomology research trials about 10 years ago, and they did exactly what I just mentioned. They had planted their field trials immediately after glyphosate treatment, and the Rhizoctonia came in and caused this bare patch. And you can see you're not going to get a good yield out of that. Okay, the last group of targets for seed treatments are the root and crown rots. The primary organisms involved are Fusarium and Cochleobolus, and I had a graduate student in 2009, 2010 who pestered you guys and in 10 other counties to do a survey on these pathogens. And what we found is, well, they're both pretty common, but Cochleobolus is more common. But Fusarium, when you have Fusarium, is more severe. So 57%, over half the fields are affected by fusarium, 93% affected by cochleobolus. 12% of the fusarium fields have infection rates that cause yield losses in excess of 20%. Cochleobolus has much lower losses typically, but it's much more common. As Uta explained, fusarium crown rot is primarily associated with this discoloration, solid discoloration that extends 1, 2, or even 3 nodes above the crown. It is associated, it has to have early season infection, and then usually of the lateral root, and then it moves into the crown. And then a physiological stress caused by going into flowering and water stress allows the pathogen to invade this lower stem. That's in a good situation. Common root rot is caused by Cochleobolus sativus, and this is the spot blot pathogen that Uta had mentioned earlier. It is associated with attacking the subcrown internode, and there's studies that show the length of that subcrown internode. The incidence of this disease is proportional to the length of that subcrown internode, so it's kind of an interesting thing. And we'll talk about that in a minute a little bit more. We have done numerous studies on seed treatments for control of fusarium crown rot, Cochleobolus sativus. This is just one of many. What we consistently find, the products that work best on these are the Evergol Energy product. And you can only get this product through a professional seed treatment. 2 other great products, though, that work equally well is the Cruiser Max, the Cereal Max product by Syngenta that has Vibrance in it. The Vibrance product really provides excellent control for these 2 diseases. And finally, the other Bayer product, the Raxil Pro MD products also do a really good job. So here you can see the non-inoculated is getting 35 bushels per acre. The untreated control is 31. And these products are actually increasing yields significantly above the non-inoculated control. So they're controlling native problems as well. So products that I think are good for treatment of crown rots include the Evergol Energy product, Cruiser Max Vibrant cereal products, and the Raxil Pro MD products. These products have the most efficacy. Other products that are notable, Rancona does provide some pretty good responses. And there's a new product out by BASF, and we've only tested it once, so we don't know. The initial results are promising. That product, I think it's pronounced Relania, produced by BASF. I hope they change the name. I hate having to pretend I'm French or something. Anyways, we don't always. So 3 out of 4 years, we get a really good response from these crown rot seed treatments. But once in a while, we get nothing. And we believe the reasons behind that and why growers don't get them are threefold. One is typically for us, what we find is those tend to be years where we have a prolonged wet spring. And so these seed treatments provide a protective window of about 3 weeks, 2 to 3 weeks. And if the wetness around that crown extends beyond that 3 weeks, then they fail. 2 other situations that I see in growers' fields is the buried crowns and seed and seed treatments, having that seed and the seed treatment distant from the crown of the plant. And let's just talk about how that happens. You guys know your plants very well. We plant the seed, it produces its initial roots, and then it sends up a coleoptile that reaches for the soil surface. Once it reaches the soil surface, the plant produces its true crown, and that's where we get the crown rot. And that intervening space between this is the subcrown internode. Most wheat cultivars are okay at getting to that surface. We want that crown to be formed at about a ½ inch below the soil surface. And most varieties are just fine at getting up there as long as this seed is planted at 2 inches or less in depth. But if things happen and, you know, geez, well, you know farming, this seed ends up getting at a deeper depth. About 3/4 of the varieties out there right now just don't have the capability of getting that crown up to the soil surface. And so you have an issue there. The main target, if you want to make seed treatments work and you want to manage crown rot, the big target I look for is trying to get this crown being formed at about a ½ inch in depth. And if you're planting below 2 inches, then all of a sudden your seed treatment and your seed is considerable distance from your crown. And that's, we're really wanting to protect the crown. We're not trying to protect the seed when it comes to crown rot. And so planting below, at depths below 2 inches is something we really need to work well. And actually most of you guys do work really well on it. And then the other thing that we see oftentimes is furrow closure is an issue in really heavy soils. And these two samples here just demonstrate that. This grower planted actually, his target depth was about 1.5 inches or so. And it was in a very heavy soil and his packer wheel was not closing furrows. So when his equipment was planting at about a ½ inch depth, he had great stand and the plants looked great. But when he planted at the 1.5 inch depth, those, with the furrow being open, the crown is being formed in the bottom of that furrow. And this whole stem tissue is just highly susceptible to the crown rot and common root rot pathogens. And so this is a big issue well, there's nothing good that we can tell you about that, but getting your furrow closed is an important part of it. I came up and saw you guys, not this summer, but the summer before. We are developing a line for spring wheat that you can plant at 4 inches depth and it's going to produce your crown at a ½ inch depth. So I'm trying to get the spring wheat breeder to move this forward and have it released in the next year or two, so that we are finding without problems with planting equipment that this genetics increases yield by about 7%. So we're hoping to full forward with that product going forward. Let's talk about application of products. We did a drill box survey with a number of extension, using help from a number of extension agents, where we went out to drill boxes, growers in the field, grabbed some of their seed, and then we did a fusarium greenhouse bioassay to test their seed to see how well their seed treatments were working. And what we found was, and this is information I probably presented to you guys before, is on-farm seed treatments. You guys are doing a great job, as good as the professional guys. Sometimes in some counties, the professional guys working a little bit less. Sometimes the on-farm guys are working a little bit less, but on average, there's no difference. If you can get your system working well, on-farm seed treatments work great. So we're getting an average around 85% emergence and survival of seedlings for on-farm, and similarly for professional. Our inoculated control, untreated seed treatment had 46% survival. So the seed treatments work really well on controlling these things, and on-farm treatments, you shouldn't shy away from doing it on-farm. The cases where we had the low performers were usually either the grower used a lower rate, or they used a lower volume. And when you use that lower volume, your grain will have this speckled appearance, and we find that associated with poor performance of your seed treatment. What we're really after is on-farm seed treatments is we want our seed to look more like it's stained. It's still going to have a little bit of a speckled appearance, unless you're going to immerse it in the stuff. You're still going to have a little bit of a speckled appearance, but it's going to kind of look more like a stain than a painting or a speckling. Using color to see if you got good delivery of product is just not a good indicator, except this particular case. And so I guess I say, you know, if you're going to use your seed treatment for protection when you're going out deer hunting, you probably use too much. And in this particular case, the grower had a decimal error and applied 10 times the amount of seed treatment that was necessary. And seed treatments applied at high rates are phytotoxic. He got less than 1% germination, while the average was 94%. So that's bad news. We looked at systems. We found no difference between the different systems in the effectiveness of application. With one exception, we had some problems with a couple of seed treatments were causing the auger slurry systems to get clogged with the seed treatment product. And I think those issues have been solved. So that stuff's working really well. Let's talk about the benefits and why you want to use seed treatments. We did a study in 2019, and the results I present here are similar to what you see with the agronomy studies, where they're looking at seeding rates. What we did is we used a seed declare, and we seeded at 5, 10, 15, up to 40 viable seeds per square foot. And that reflects going from 200,000 to 1.7 million, or about a quarter bushel to almost 2 bushels of grain per acre application seeding rate. And then we use no seed treatment. And this is not in a field where we inoculate or anything like that. And what we observed is that we got in this field an actual stand that was about 50% of what we seeded at. If you're using a seed treatment and you get that, you should change your seed treatment. In fact, if you get anything below 80% of what you expect in terms of emergence, you should look at other seed treatments. Seed treatments there's a lot of good seed treatments out there. They're not all the same. Some of them will work better in some people's conditions than in others. So this green line is the expected, and this is what we actually got. As I mentioned, there's a number of agronomy studies looking at seeding rates, and probably 2/3 to 3/4 of those studies are reporting, when they do, they report stands around 50%. So it's not an uncommon thing. And when I go out to growers' fields, if it's spring, I ask them what their seeding rate is, and then I go out and count. And I won't say this always happens, but I see probably 1/3 of the growers' fields will have, if they're not using a seed treatment, they'll have a 50% stand. So you guys know what your sweet spot is in target for stand. For me, in Bozeman, I start seeing yield potential going down when I get below 15 plants per square foot. That's when I really start seeing the yield potential going down. And that's, that relates to a lot of other things too. So if you're planting early versus planting late, you may want to adjust your seeding rate. For me, this is where that's when I start looking. But I only plant an acre, and you guys plant a little bit more than that. So there's only two published studies that look at the yield effect of seed treatments. One was done by Dick Smiley he's in Oregon. It was done in 2011. He did his study using a number of commercial fields in Idaho and Oregon. This is hard red spring wheat. He used a dual seed treatment of Vibrance and Rancona. mix. And that, because he basically seed treated twice, it was pretty expensive seed treatment. It cost him $11 per hectare, which would be about $4.50, $4.45 per acre for you guys. What his benefit was, there's a lot of noise in that system, but statistically it was significant yield benefit from this treatment. He gained 419 kilograms, and he estimated the economic return was 10 to 1. He got $100 per hectare extra from using a seed treatment. So just to give you an idea. I worked with Peggy Lamb and Greg Carlson, God bless him. In 2010, we did a similar thing going into a summer fallow field for winter wheat. And what we found was the better products. This Syngenta RCA1, by the way, is actually the Vibrance and Rancona mix. And Bayer Proseed and this BSF product, particularly the Stamina product, those perform better than untreated check at a significance level. And so all of these products are between 2 and 4 bushels gain in yield per acre. And so I usually say the break-even point is 2 bushels for seed treatments in winter wheat. But this is sparse information. And so that brings me to this ask. I've been up at Phillips County. I've been with Marco. I've come out and seen you guys. And I've had a couple of you guys complain about, spending $700 for a 2-gallon jug of seed treatment. And it's a valid question. We don't have good information about what the economic benefit is for spring wheat. And so I recruited Tom Grupp. Tom is from a farm family in Denton, Montana. He's a crop science student. He did an undergraduate project for me, looking at bin seed and pathogens associated with bin seed, because most of us use bin seed. Even I use bin seed for my research. And what he had found with that bin seed survey down in the Denton area, looking at bin seed from his neighbors, was that 80% of your bin seed is contaminated with Cochleobolus sativus, common root rot pathogen, spot blot pathogen. And 30% of the seed itself is infected in those batches. So it's a very common thing, and it would kind of explain why common root rot is so common. So anyways, this January, just a couple weeks ago, he's starting his project in plant pathology, looking at the benefits of seed treatments and seed-borne pathogens. And so he has 2 parts of it. One is to determine the benefits of fungicidal seed treatments in spring wheat and winter wheat production systems. And this is not me doing trials in little plots. I want to do this in commercial settings with you guys, so that we actually have data that is most valid to you. And then we also want to survey pathogens associated with seed. The ask we're trying to make, and I've discussed it with him, and I've talked to a number of other growers, is we want to recruit participants who are already going to use either Evergol Energy or Raxel Pro MD or Cruiser Max Vibrant cereals in their production system. And our goal is to have at least 12 for spring wheat and 12 for winter wheat. And all we're asking of these people, I'm a little nervous, I can't see your faces, guys. All we're asking is that we're looking for somebody who's willing to plant a strip without the fungicidal seed treatment. You can use an insecticidal seed treatment throughout everything, but we want people to go out there and plant a strip without the fungicidal seed treatment, and then we want them to be able to go in there and harvest that strip and harvest a neighboring strip with the seed treatment and take it to the elevator and get it measured to get an accurate yield estimate for both. And then the last thing we're asking is that you let Tom visit your fields and make observations on the stand benefits and the relative amounts of diseases, particularly the crown rots, And even we also suspect that there may be a benefit with regards to weed suppression as well. If you're willing to participate, we have 5 spring wheat growers already willing to do this work. Please contact Tom at Thomas Grubb at student.montana.edu or talk to Marco. Marco begrudgingly, such a nice guy, Marco, and Kate Hatfield and even Uta have all been willing to participate in this project. So we really would like to get the best answer we can for whether seed treatment is a good investment for you guys. The other part of the project is that we will be going with extension agents to collect bin seeds from various counties throughout the state. And we will be looking in those cases, we'll be looking at pathogens associated with seed. Is Cochleatus sativus as common throughout the state as it is in the Denton area. And we will also be examining the emergence of that in seed and commercial seed as well. We'll use commercial seed. And if we do have pathogens, what we will be doing with those pathogens is looking at whether they have resistance to some of these fungicides that we use as seed treatments. So with that, I'll take any questions anybody has.
Questions for Dr. Dyer?
Can you hear me?
Go ahead.
There's more plantings of winter wheat forage crops like willow crick and ray and that kind of stuff. Is there any kind of research being done on those particular varieties, or even winter triticale? Just curious.
I haven't done. It's a really interesting area and it's a growing area. I haven't done any research on them at this time, but they are definitely something that's of growing importance. But at this point, I haven't done anything on them.
Okay, thanks.
Yeah. Tim, any question?
No, no, we're paying good attention and looking through everything, but no questions.
Okay. I hate
Slide comments without the mic muted, but no questions.
this video thing is just killing me. So I like to see you guys' face and talk to you guys. So. Okay, well, I appreciate everybody's time. I hope that some of this is informative to you. And if you're interested, well, if you're willing to sacrifice a little bit of your time and your acreage to do a study to understand whether seed treatments are an economic good investment for spring wheat or winter wheat, please contact me or Tom about that. And we really appreciate it. Thank you very much.
Thank you, Dr. Dyer. Appreciate it. And we look forward to seeing you maybe this summer, if possible. Wait, we might have a question here.
I'd love to come up.
So if you are online and you're a grower, if you can look in the bottom, there'll be a little box right in the middle of your screen that says chat. Please respond with your name and your county, and I'll print that out or save that file so that we can get you the credits. For most everything, you'll get 2 credits for attending, except seed treatment. If you're a seed treatment person, you only get 1 credit. That's how stingy the Department of Ag is. Or did I say that way? Sorry. So I didn't see any questions, but Uta and Dr. Dyer, look at the chat in case there are questions that you can see. I didn't see any. So I'm sending around your pesticide deal. If you don't have your license number with you, don't worry, we're the government, we can find that out.